Great, thanks. Thanks so much. So again, I'm Kevin Drew. I'm an assistant professor at the University of Illinois at Chicago. First, just want to thank the organizers for giving me the chance to tell you a bit about my work today on essentially discovering and identifying direct interactions between proteins in the cell. So just to make sure that we're, I guess, all on the same page, proteins are the functional unit of the cell, but they also don't work alone. They work in concert with other biomolecules. And so we can just look at a handful of essential functions of the cell, like replication, transcription, translation, degradation, and we look at the proteins that are actually carrying out these functions. We see that there are many different proteins that are self-assembling into these large macromolecular complexes. So we see RNA pull in the mediator complex, we see MCM in the OR complex, we see the proteasome, we see the ribosome. So many different proteins self-assembling. And what's really nice about these examples is these are really well characterized examples. We have protein structures for these complexes, as well as since we have protein structures for these, we can really understand the molecular mechanisms of these functions. Okay. But we don't have this for all protein complexes. And so really kind of a driving approach to my lab is really, can we generate a map of all physical associations in the cell? And I like to kind of think about this akin to, say, Google Maps for the cell, where we can kind of have this very broad view of what the cell looks like and then be able to zoom in. And this is sort of at this level here in the middle here is something that Anne-Claude had kind of touched upon. And then where we can get proximity and organelle level. But we also want to zoom in even further and be able to identify and really understand biological entities such as protein complexes. And so to get at this level, we really need to know all of the components. We need to know the RNAs, the proteins, the biomolecules that are making up these complexes. But further, we also want to essentially pick up our little person and drop it onto a roadmap on the cell and be able to walk around and look at these complexes, really understand what they're made of, and how they're assembled and who's connecting to who. And in order to get to that level, we really need to have structures for complexes, these structural models. So in previous work, we had integrated high throughput proteomics experiments, affinity purification, cofractionation, and many others to build a set of protein complexes, which we call HuMap. And so we're identifying over 7,000 protein complexes. We're covering about half of the human proteome here. And what I'm showing here is a cytoscape network where these sub-networks represent protein complexes. And each one of these is backed by some experimental evidence that we can go back and track down in terms of the proteomics there. And you can see the web link there if you're interested. But really, we want to get to the street level of protein complexes and really understand what their molecular mechanisms are. And so we can really ask this question of what are the structures of these 7,000 complexes. And we're really at this amazing time in structural bioinformatics right now due to the advent of AlphaFold and AlphaFold Multimer and a lot of other tools that are available where we can give it a sequence or multiple sequences and then predict the three-dimensional structure of these complexes. But we really need to understand what proteins to actually give to AlphaFold. We can't. There's 200 million possible pairs of human protein interactions, and so we really have to ask this question of what pairs of proteins are we giving AlphaFold? And so this was addressed by Pedro Baltrao and Arnie Alfson, among others. And essentially, they built a large compendium of AlphaFold multimers, and they used HuMap as a ranking, so high confident rankings of HuMap pairs to then give to AlphaFold. And this actually worked extremely well. But what's interesting about HuMap, it wasn't designed to do this. This is really a co-complex interaction. If you're in the same complex, you're drawing an edge, but that doesn't mean that you're directly touching and physically interacting with each other. So really what we need is a model to discriminate between direct and indirect interactions. So here I have a structure of ARC23 on the left, and we see it as many different subunits. Some of the subunits are actually directly interacting, physically touching, such as the cyan and green protein. But we also have other proteins that are indirectly interacting. So the magenta and the green protein, for example. And so really what we're interested in is being able to discriminate between these direct and indirect interactions. And we've shown that cofractionation mass spectrometry, and Claude gave a nice introduction to cofractionation. But cofractionation actually has some structural information in it. And so if we look at just the proteasome, subunits of the proteasome, fractionated based on ion exchange here, as we see nearly all the subunits of the 26S proteasome are all colluting, but we see the proteasome subcomplexes actually have different signatures from other ones. And so the base, the lid, and the core all essentially are fractionating separately from the full 26S. Okay, we can hierarchical cluster this and actually identify direct interactions and build up the structure of the proteasome this way. Okay, in a previous work, we actually integrated a lot of this cofractionation data using a graphical model approach, and this helped us identify direct versus indirect, and we were able to build structural models based on this. And for example, this Cheops complex here, which didn't have a known structure, and this is all pre-AlphaFold, and this has been holding up. This model has been holding up since then. Okay. And so now we want to advance this even further. There is over 25,000 mass spec experiments now, many from cofractionation mass spectrometry, but also from affinity purification mass spec experiments. So the Bioplex group has thousands and thousands of pull-down experiments in which they have some structural information. And then there's other data sets such as RNA pull-down experiments, as well as the proximity labeling that you wouldn't necessarily think that has information about direct interactions. But we can reanalyze some of these, similar to what Anne-Claude talked about in terms of these prey-prey interactions. And that actually has a little bit of signal for direct protein interactions. So we can build a feature matrix based on this. We can label it with direct pairs from the protein data bank using Interactome 3D. And then we use a model selection algorithm called AutoGluon that can then build a high confidence model prediction. We can then run all of our pairs of proteins and ask, are they directly interacting or indirectly interacting? We can do this for over 80,000 protein interactions. Okay, so just as an example of a large protein complex, how well are we doing? We can look at the CCT prefolding complex. So this is a large macromolecular protein complex, many different subunits. Many of them are directly interacting, but also many of them are indirectly interacting. So how well are we actually discriminating between these two? And so on the right here is we have the direct contact score, where 1 is very confident, 0 is less confident. And we actually see fairly good discrimination between our direct and indirect interactions here. So in green are directs, and gray is our indirect interactions. Okay, we can just look at the top 5 scoring predictions here, and they're all directly interacting. And not only that, is we look at the structures of these, and they actually have a lot of surface area interacting surface areas at these. So this suggests that we're doing something right here. Okay, we can look at this on a leave-out benchmark in terms of comparing this to other models, and we see that we're outperforming these other models, such as my previous HUMAP2, but also the previous direct contacts based on this graphical model approach, as well as a used-to hybrid network, the HURI network. Okay. The other thing that we can do is we can look at this compendium of AlphaFold models that were created by Pedro Beltrán and Arnie Elfson's groups, and we can essentially evaluate the AlphaFold 2 model confidence. So they do this based on P.Q, which is this x-axis here, and we see this bimodal distribution. We see many different subunits have this low confidence. But many also have this higher confidence, greater than say 0.5 p.q. So we can zoom in on those models there, and we see that direct contact stew is enriching for this and outperforming these other models. Okay, so so can we actually build structural models based on these direct contacts? And so we can look at the intermediate filament complex. It's involved in the cytoskeleton. And we look at the top interactions based on direct contact scores, specifically between NEF-M and VIM, as well as NEF-M and NEF-L. And what we see is we can go looking for additional validation in cross-linking data, and it's supported by cross-linking data. We can also build structural models based on these. So NEF-M and NEF-L, we build a high-confident structural model based on these. And many of the cross-links are satisfied. So 6 out of the 9 cross-links are satisfied. We can also look at this NEF-M VIM model, structural model, and we see 8 out of the 8 cross-links are satisfied when we include this dimer of dimers. So we can build up these confident structural models based on these. So the next thing that we're interested in is, can we interpret mutational data with our structural models built based on these direct contacts? And so OFD1 is a protein, is a ciliogenesis factor that recruits other ciliogenesis factors, and it's involved in docking of the cilia vesicle, which then goes on to interact with the plasma membrane and extend a microtubule structure protrusion during ciliogenesis. Okay. When OFD1 is disrupted, when it's mutated, it doesn't go through this process. It doesn't recruit these ciliogenesis factors, but instead gives rise to these clinical phenotypes in patients. So you see a syndrome called oral facial digital syndrome, and I'm showing an MRI of a brain malformation in some of these patients. Okay. So can we start to understand some of these mutations in a structural context? We know that OFD1 is actually part of a oral facial digital syndrome complex. It's predicted by our HuMap2. And so we can take the high confident direct contacts from this complex and identify OFD1 is directly interacting with FOPNL, and FOPNL is directly interacting with this KAO753. And there's many other subunits here, including KOS753, that have patient disease mutations. Okay. So we can build structural models based on these, and we see that they're very high confident. We see OFD1 FOPNL has a very high confident P.Q score, as well as this FOPNL KO753. And then what we can do is map these mutations from OFD1, and we see that they're actually clustered together in the structure, but not only clustered together, they're clustered at the interface between FOPNL and OFD1. So these models are able to give us some structural context in terms of the molecular mechanism of these mutations. So in total, so here's the full protein interaction or direct physical interaction network of direct contacts too. We have 15,800, roughly 800 high confident interactions. We're covering about close to 6,000 complexes that have one of these high confident interactions. So you can start to build structural models of these complexes. 440 of these are supported by cross-links in a large-scale cross-linking experimental data set, as well as 1,500 of these are supported by high-confident AlphaFold 2 models. Okay. And so with that, I just wanted to thank some of the members of my lab that worked on this. So Aaron Clausen worked on this project for a while. And I'd like to thank funding and appreciate your time for listening.
Thanks, Kevin. We have time for a few questions.
Hi, I'm not a structural biologist, but I've been wondering, do you think that based on this binding and how strong this is, we can say something about how long these protein complexes last in the cell, and perhaps even say something about how far they can travel in the cell when they're attached to each other?
Yeah, so that's a great question. In terms of binding affinity, this is there's a lot of different groups that are thinking about protein interactions and binding affinities, being able to computationally predict these things. I actually have another project in my lab where we're denaturing complexes using urea and getting affinities and things like this. One thing that you traditionally like to think about is just the surface area. The more the surface area, you'd expect a higher binding affinity. Whether that holds up in terms of some of our experiments and things like that, it's still preliminary. But really determining what are the factors that are governing these interfaces and the high affinity interfaces is really quite an interesting topic. Absolutely.
So I had a question about improvement. So can you talk a bit about, are there trends to where you don't predict well? The direct interactions, small complexes, large complexes with a lot of potential indirect connections. And what data do you need to make it better?
Right. So in these experiments, you definitely need things to essentially fall apart. That's really where if we kind of go back to the cofractionation experiment, you really need something to fall apart to see these signatures that it's a subcomplex. So actually, we do fairly poorly on the core of the proteasome because it never breaks up. It's extremely stable. The other trends that we see are just the amount of times you see it in these experiments. And so really it's just an abundance. We do better on more abundant protein complexes than others. And as we sample more and more, we're going to do better. Mm-hmm.
Kevin, I was curious, like how do you about the yeast to hybrid data? I would have expected that would have done particularly well for direct interaction. So the fact that it doesn't seem to do as well as what I would have thought, is it a coverage issue or false negative issue, you think? Or
right. So this is sort of perplexed us for a little while. I really think it's a false positive, false negative issue. When we look at the yeast to hybrid data, and you'd expect to see pairs of proteins that are directly interacting in complexes like the proteasome and things like this. We don't always see them in the yeast to hybrid data.
Sure.
And it's also many false positives interacting with other things. So we haven't really been able to trust the yeast to hybrid data as much as we expected because it's really assaying this exact interaction. So,
thank you.
yeah, it's a great question.